so today we are going to finish our chapter on Darwin and descent with modification. Now, I want to warn you at the beginning of this video, there are some pictures in this video that are slightly graphic. Nothing like bad, just they're of some different infections, and I'll warn you guys before I get to the pictures, just in case you want to look away, and then I'll let you know when they are done, so that way if you don't want to look at them, you don't have to. They're slightly gory, okay? So let's go back to talking about evolution, adaptations, that type of thing. So there was a study done in Florida, different parts of Florida, about soapberry bugs. So what they have these beaks, and they can eat different seeds inside of fruits. Now, in South Florida, they noticed they have longer beaks because the fruit are larger. In Central Florida, they have shorter beaks because not so much of the big fruit there. So here's what they look like, kind of like a stink bug almost, but a little bit different. So the beaks have evolved with our soapberry bugs in the different parts of Florida. And this actually happened really quickly because they were able to reproduce very quickly. So the faster an organism can reproduce, the faster evolution can occur. Now, in Central Florida, which is the green bar graph here was the beak lengths for different soapberries that they got. And the orange one up top is going to be in Southern Florida, where the fruit is a little bit larger. So we can see that the beak size is completely different. And in Central Florida, there's no reason for the long beak ones to be there because they can just go south and have the fruit they like. Same thing with the short beaked ones from Central Florida. They can't be in South Florida because they're definitely not even going to get the fruit. They're not going to be able to get their beak in to get the seeds, so they would naturally die in that area. Okay, so we're going to talk about a bacterial infection called a staph infection. So these are on people's skin. I've actually seen a person with a type of staph infection, and I got to drain it, which was something very interesting. But so you have an infection, and just like with any infection, you're going to have some swelling in the area. You're going to have some pus and other things in the area, which is going to be your body trying to fight the infection. But they found that some strains of staph infection were actually resistant to certain antibiotics they were giving. So usually methicillin here is a drug they can use to treat bacterial infections. So when they were trying to treat it, they found that for a while, yeah, it worked. Everything was good. But after a while, they found that any kind of psyllin, so methicillin, any kind of psyllin, penicillin, wasn't working on this one staph infection called MRSA. So now they are resistant, and if you use a psyllin on MRSA, not only is it not going to kill it, but it actually makes the MRSA grow and reproduce faster. So if you have MRSA, you never want to use a psyllin on it. So let me show you what MRSA is. Pictures are going to get not graphic on the first one, a little more graphic on the second one, and the third and the fourth one, definitely graphic. So if you want to fast forward about 30 seconds, you can, just to not look at the pictures. So here is our first one. This is just MRSA, okay? It's under the skin. It's very early MRSA. A little bit further into it, all right, the MRSA is starting to eat away at the skin. It's underneath. Now, if you let it go long enough, here's the graphic pictures. All right, it can actually really tear your skin apart. And the last one, even worse. Yeah, crazy, right? So, pictures are all gone. I told you I was going to try to be quick with them. So, you really don't want to play around with MRSA. I know a while ago in a school in downtown, so far away from us, like it was like an hour away, there was a MRSA outbreak, and they actually shut down the school because it was so serious. They didn't want to take any chances with it. Over time, we have found that there are actually more MRSA cases in hospital because it's continuing to get resistant to our antibiotics. Good news is we don't see it very often. All right. Okay, getting back to natural selection. Natural selection does not create new traits. Mutations can. All right. People moving around, bringing traits to different areas, that can change the gene pool. All right. But natural selection does edit what traits are going to be more prevalent in a population. All right. So that's what natural selection does. We already talked about evolution, its descent with modification. We're going to go over some structures now, and I'm going to show you guys how they relate to evolution. So our first one is homologous structures. Homologous structures are limbs, things like that, just body parts that are very similar in other organisms because we share a common ancestor. So let me show you what's going on here. So for homologous structures, we are going to have a common ancestor. We're going to share a common ancestor. However, even though they may look the same, okay, so the internal structure may be the same. I'll just put as similar. The function of this structure is going to be different. So similar structure, but a different function. Similar structure, different function. That is a homologous structure, okay? So an example of this, if you take our arm, whole arm, okay, from where it connects to your shoulder all the way down to your fingers, and compare it to a cat, a whale, and a bat, the bone structure and where the bones are going to be very, very, very similar. This tells us that we probably shared a common ancestor, maybe way far back, maybe sooner, with these three organisms, just because the bone structure is the same. You guys can see the internal structure is very similar. However, the function is different. We use our arm much differently than a whale uses its flipper or a bat uses its wing. Cat, kind of similar, so that tells us that we are probably closer on the evolutionary tree to cats than we are with whales and bats. Second one, we're going to go over vestigial structures. So vestigial structures are remnants of structures that maybe serve different functions in our ancestors. So maybe it was our species, maybe it was a different species that we descended from. All right, let me go over this example here. So in this example, wisdom teeth, yeah, we get them removed. We really don't have wisdom teeth, or we don't use them anymore, and they create more of a problem for us. In this, the coccyx, which is our tailbone, we maybe once had a tail, and that tail's no longer there. Okay, so yeah, that's the remnant of what our tail might have used to be. This here, the lacrimal caruncle, this is right here on your eye, okay? It's that little piece right there of flesh. Now, we still speculate this is a vestigial structure, but it's kind of turning into its own little structure. Let me explain. We think it's vestigial because we think maybe at one time we were underwater, not us, just a previous past common ancestor. And we think that was our third eyelid to help us see underwater. Now, we don't really use that anymore, so it's a vestigial structure. However, we still have it. Why do we still have it? Well, we think now it's actually used to collect sleep and all the little crusties in your eyes, when you're sleeping. So that's why we still have it. Last one over here, whales. And we'll talk about whales a little bit more, but we think that this little bone right here, which serves absolutely no purpose, it's the femur of the whale, that bone once was a leg. connected to a leg. And you can see it's still connected to kind of a floating pelvis there. So we think the femur was once a leg for a whale, which now they don't have legs anymore. So that's a vestigial structure as well. Okay. All right. So with our genes, we share a lot of the same genes, a lot of the same DNA with every single organism in the world. So we might share genes with bacteria even, and bacteria is like way, way, way far away from us. What this tells us is that our genetic code for all organisms is very similar, and we probably descended from of these same organisms, since these genes not only have the same pattern, ATGC, but they also encode for the same proteins, for the same genes, okay? All right. We're going to go over two different forms of evolution here. First one is called convergent evolution. So if you think of something converging, it's coming together. So we're going to have a last one called analogous structures. So let me write that one for you here. So if you remember homologous structures, keep those in mind while I write this. Analogous structures. Okay, so first thing, they have a common function. 2, they do not have a common ancestor. So this is the exact opposite of homologous structures. Homologous structures, we shared a common ancestor. The internal structure was the same, but the function was different. Analogous structures, they have the same function, but they don't have a common ancestor. That tells us it's going to look the same, but and it's going to function the same, but there's no evolutionary common past between these two. So what's happening with convergent evolution, guys? We have two organisms that are going to look the same. Okay? They're going to look pretty much identical. However, they are going to share no past. Their past is not going to be related. So why would we have these? How did we get these organisms? Well, maybe they're on two different continents, and this is usually how it occurs. They're on two different continents, and that land, that ecosystem, has a need for a certain organism. Evolution will take effect here, and they will actually evolve. A species will evolve to fill that missing part of the ecosystem, to fill that niche of the ecosystem. If you look here, sugar gliders, all right, kind of like flying squirrels. We have some in Australia, we have some in North America. They look almost identical, but they share no common past. That is convergent evolution, all right? So convergent evolution, they're not related, but they look the same. So for our fossil record, we are able to take different parts of an organism, and when we have these different parts of an organism, we can compare them, the structure of them, the parts of them, how they fit together with others, and see, okay, is this going to be related to another organism, or is it not going to be related to another organism? So our last part of this, before I go over an example of how to do a cladogram, is just going to be, we're going to go back to the whales thing. So whales transitioned from land to sea a long time ago. Now, if we look at this picture, we have the whales right down here, and we have a bunch of different organisms up here. So the whale went from land to sea. If you guys have ever seen, baby whales, they actually swim on top of the mother. So that way, when the whale needs air, because they do need air, they don't like breathe with gills like regular fish do. the mother will actually slowly lift itself up to the surface, so that way the baby can get up to the surface and take a nice big breath, right? Because when they're young, they actually don't have enough strength to get up to the surface of the water. So they once started on land, and they once had 4 legs, which we talked about with the vestigial structures. Now they don't have legs anymore. So why do they not have legs? Where did they go? Well, we already talked about they were on land, they had 4 legs, now they went to the sea. Since they were in the sea, they didn't need the back legs anymore. They didn't use them. So the back legs were actually a greater cost than a benefit, because they still had to pump blood to that area so the legs didn't die. So over time, they actually lost the back leg structures. Okay, so let's go over an example here. This example, give me 1 second, guys. All right, this is a past AP question, and I'm going to show you guys how to set up a cladogram. A little bit different than the ones that we've been working on. But I have 5 different species here. So we have the genus name first, species name second. We have like E. ferus all the way over to E. africanus. What they did was they showed us the number of amino acid differences between these 5 species. So let's make a cladogram or a dichotomous key or a phylogenetic tree, however you want to say it. Now on the AP test, they started giving you this. So here's going to be our first step. We are going to find the highest number, and I put the rest of it down here. We're going to put the highest, get the highest number right there, and our goal is to try to put in the species here. So we got our highest number, which is 21 in the whole thing. And the next question is, okay, we got E. ferus and E. polylepis here. One's going to go on one side, and one's going to go on the other side of our cladogram. How do we determine which one? Well, I'm just going to randomly do it here. I'm just going to say E. ferus here. And on this side, I'm going to say E. polylepis. I might be right, I might be wrong. If I'm wrong, I'll switch them. So we got that number done, 21. Okay? So let's see what's going to be next to each one of these next, the next bar over. So E. ferus. The next one over is E. africanus. So the next closest one is going to be E. africanus. And that tells me from here to here we have 1 different. Okay, so that distance is 1. E. polylepis here. Okay, so we did that one already, 0, 0. That's the same one. It's 17. E. forsteri. That means this is 1 and that's 17. That doesn't make sense, okay? So what I got to do is I got to switch my first one. So what I want to do, guys, was show you how you make sure that you're not making a mistake. So E. ferus is going to be on this side, and this side is going to be our, I'm sorry, D, not E. E. polylepis. All right, now we can do the rest of it pretty easily. We know the next closest one to E. ferus is E. africanus. All right. Next one over, I'm just going to use the E. ferus one. So we did that one. Next one over is going to be A. forsteri because it's 13 away. I'm sorry, I wrote the wrong one there. It's going to be G. gallus, or G. gallus, because it's 11 away. My fault, guys. Numbers are small. I thought it said 17 at first. So the last one is A. forsteri. And we could check. D. polylepis here. If we go over, its next closest one is going to be A. forsteri, 17. And then one more over from that, a little bit further, should be G. gallus. Okay, so that's how you set up a phylogenetic tree or a cladogram. All right. All right. Last 2 or 3 slides here. All right. So one time we thought that, okay, we had all our continents and they were always like that. We found that actually wasn't like that. Originally we had a one big, gigantic supercontinent called Pangaea. And what happened was that Pangaea slowly broke apart into our other continents, and they moved apart slowly, and they're still moving apart slightly to this day. And that happened, I mean, it took millions upon millions of years for this to occur. But we had one big supercontinent, and then we got our individual continents now. So endemic species, these are species that are native to one area of the world. They're not found anywhere else. Islands are a really good area to find these because if it is trapped on an island, okay, birds aren't going to be like this, but if they're trapped on an island, they can't get off, and it's not like they're going to go off the island, breed, and then come back. They can't get off the island, so they're going to stay in that area. The only way that could happen would be if humans, starting zoos and stuff like that, we took them off the island and we took them to the zoo and started to, make them reproduce in captivity. An example of this is lemurs in Madagascar, okay? Those lemurs are only in Madagascar. Tortoises in the Galapagos Islands, they are gigantic. I don't know if you guys have ever seen a Galapagos tortoise, but, I'm not afraid of turtles, but these things are huge. Like, they're like half of my body size, all right? Okay. All right, guys. I don't have to go over anything on this one. So that's it for the chapter. You guys can go ahead and finish your reading guide, and have a good rest of your day. I'll see you next time.